introduction. What I will try to do is to show that many sciences these days should come together and create new knowledge, and this society is about new knowledge, because so many things we can do not only within the boundaries of your own research, but when we exchange our sciences, our knowledge, and bring together biology, material science. I'll give you some examples. We'll see how many I will be able to present, and to think how that can change life and make new, better materials, better structures, better architectures. And to begin, let's say we want to dream. And if we're dreaming and we think about imagining new technology, so what would it be that we want to have in our life? Let's say it would be materials, devices, architectures. You can help me even to build this list. We want them to adapt to their environment. We want them to harvest energy. We want them to self-heal. We want them to reconfigure maybe in some more useful organizations. We want them to collect and deliver water, self-clean, whatnot. There is so many things that one can put on this list. What I will try to convey here, that at least I feel that a lot can be learned if we think about it as a vision, building as organism. And when I say that, I would say that it's very easy to think about the first part of this list, which is to search for biological systems with interesting properties. Anything in biology is interesting. More difficult is to understand what are the mechanisms of their formation. What is it that we can learn from these biological systems to use to our advantage? It doesn't have to be as sophisticated as biological systems. I don't think we're there yet. But then, hopefully, by using this multidisciplinary approach, we can apply it to novel materials, novel medicine, and so many different things. Since there's very limited time, I will today limit it a little bit, although you will see there's a lot that I want to put in this talk. And I will limit it to somewhat plant-inspired materials. I'll talk about lotus leaf, pitcher plant, Venus flytrap, iridescent seeds. We'll see how far we will go in this list, just to show the breadth of sometimes strange ideas that can come up, that we can come up with if we think about biology as a driver for new technologies. So, chapter 1, lotus leaf. Now, we all know how clean and wonderful the leaf is, and the reason is that it's what is called superhydrophobic. It has a structure that hates water. Superhydrophobicity comes from the fact that it hates water, but it is structured. So water is sitting on a cushion of air, and therefore is highly mobile, and when it is picked up, and rolling from the surface of the lotus leaf, it picks up dirt, and therefore lotus leaf is always clean. This phenomenon is called superhydrophobicity, and just to show you what is behind it, let us say we have hydrophilic substrate. It likes water, water spreads. If you have hydrophobic, doesn't like water, but it's still in contact with it. If now you have this superhydrophobic surface, water cannot even stand on it. It jumps off, leaving surface completely free of water. It's an excellent approach to think about self-cleaning materials, but the problem, and I can already say, chapter 1 ends here. I'm already moving to another biological inspiration, because nature evolved it to deal with water, not to deal with really complex environments, with biological media, with icing conditions, in high humidity. Pretty much anything but not water would get you from these super non-wetting condition to super wetting condition, when the liquid, contaminated liquid, when the surface is damaged, will actually get in. The example that I show here, and I will come back to this example later again, this is morph butterfly. The color of this beautiful blue color is not pigment. It is what is called structural color. It's based on a structure. I'll come back to this later, but what I want to show here, it was optimized to be super hydrophobic. Water jumps off, color remains, and actually scales remain intact, but if it's different medium, in this case it's alcohol, it actually penetrates into the structure, becomes this super sticky condition. In this case, it's manifested by change of color, because color is now removed. So it doesn't work. And if we are to think about other sticky problems of our life, and I know that it's very difficult to read what is here, but intentionally done so, what says here is that materials get dirty. They do. And most difficult things, if we think about snow that we don't want to be collected on our cars, as you could see here, or if we want to clean our water, this contamination is incredibly difficult to get rid of. I can again make a much longer list if we think about infections. Again, sticky substrates that we want to avoid, or blood clots, and going to marine fouling. Our solar batteries, they cry, please clean me, because you develop all these technologies to work well, but if it's dirty, there is no way for them to work the way we expect. And graffiti, I'll talk about that as well. I really don't like graffiti. So we can change many things if we prevent material structures from contamination, and that will affect energy, health, environment. So what is the better strategy that we can use? And I will show you one. And in this case, it's chapter 2. It's pitcher plant. Here's pitcher plant. That is really interesting. It likes to eat these nice creatures. But suddenly, on a wet day, they cannot run anymore. They actually sliding into the digestive juices of a pitcher plant on their own. It doesn't even have to capture them. So something interesting is happening here when insects cannot run on a dry day. Everything is fine. The moment it's wet day, it's high appetite. They're running into the mouth of that plant. So what is happening here? Let's just, very short, I would not give you a very detailed description of that. Interestingly, the structure of this plant is very similar to the one of the lotus leaf. It's also structured surface. So there is a structure there, but it's now not hydrophobic, it's hydrophilic. When you have structured hydrophilic surface, it loves water. So what it does, it picks up, on a wet day, it picks up a layer of water that now covers the structured surface, and it becomes water that is seen by the oily insect feet. The outcome of that, oil-water interface, is very slippery. Everything slides. In fact, it doesn't have to be pitcher plant. Same principle is in our stomach, in the scales of fish that allow them to swim better, to protect our gastric lumen or our eyes, and this is eyes of my grandson, but there is always a layer of liquid. Let's think about these structured surfaces with a layer of liquid on them, and based on that, my lab developed what we called slippery liquid-infused porous surfaces, or SLIPs. And they're really omniphobic. They hate everything. Nothing can stick to these surfaces. The principle is kind of counterintuitive. We're using roughness to create non-rough, ultra-smooth surfaces. What happens there is that in roughened surfaces, if it's properly chemically functionalized, you now have a way to capture a lubricant. It doesn't have to be water, as it is in pitcher plant. It could be any lubricant that I can choose, and that keeps this lubricant within this porous network, which means it creates a smooth, slippery interface from which everything slides, and in this way, one can say, slippery surface. Everything slips. Let me show you a couple of examples of what is happening on these surfaces. This is PBS Nova. As I said, I don't like graffiti. That was in my lab. What is talking about that there is no graffiti on the slippery side of things. I can show many more examples. Let's remember, and it's very important, that since its liquid is now active part of the material, it means that it has self-healing capabilities. So superhydrophobic surface, crude oil is not moving because it's really difficult medium to handle, but self-healing takes place on a slippery surface. I've just shown a couple of examples. It doesn't have to be exhaustive. Here is an example of how it deals with anti-icing. The same substrate, the same cold plate. There is ice, frost actually, forming on one side, but there is no ice or frost forming on a slippery surface. Or if we think about biocontamination, this example that I show, first of all, shows that we can make them, if we want, transparent. So as half of the glass is just regular glass, and the other half is made slippery glass. And you could see this algae layer is not forming, well, it's forming, but not attaching to the glass slides. So what can we do with that? A lot. For example, if we think about medical devices, I'll just show one potential application in bronchoscopy. You need your lenses to stay clean, even if they're in contact with biological medium. For example, in this case, I show the case of biopsy performed on pigs with the slippery surface. You dip it in blood, but it immediately shows the image, so there is no contamination. You continue the biopsy procedure and endoscopy imaging. Let's think about applications in marine fouling. So here's a couple of examples of testing these materials in the form of paints. Compared to regular materials that are used now, you could see that slippery surfaces as a paint for marine application can keep the surfaces clean of fouling. And I will show just another one. So going from just demonstrations to actual applications, the one on the right is showing the application in water harvesting. And what you should see here, that this side is not just slippery surface, it's a little bit more. And I will mention now the merger between different approaches. In this case, just slippery surface will not be sufficient, but if we combine the lessons from other animals, in particular from desert beetle that develops very interesting bumps on its surface, but also from cactus that has asymmetry in its needles that allows water to be transported in a specific direction. We can design the surfaces that can collect water, and I can tell you that while this one is nothing is even happening, the amount of water that can be collected on the surface using combination of these three approaches is about 2 orders of magnitude than what can be done if we're talking about water harvesting on regular materials. Okay, chapter 2 is done. Chapter 3. Yet another interesting case. I will talk about wheat-ons, and you probably know, if not, I will tell you that they're really interesting ability to to introduce themselves into ground deeper and deeper, simply by changing an angle between the ons that change the force that they have on putting them into ground. So they see themselves better and better. just opening flowers would be another example to mention. If we think about Venus flytrap, the common feature, and if cilia and flagella has a lot in common with that, or at least the way I want to simplify these very different biological lessons to maybe a set of simplified rules that would be sufficient for me to create reconfigurable dynamic materials. So if I were to ask, what is the common feature, simplest common feature here, it is reconfigurable, moving, high aspect ratio, hairy materials that are put in motion by a muscle. So what can we do as material scientists or chemists to do that? What we can create is what we call hairs, which is hydrogel actuated integrated responsive structures. So what we actually do, we create these high aspect ratio hairy surfaces, but we place a layer of gel that acts a function of a muscle, that when the gel, as it responds to external stimulus, contracts, the fibers go down. When it expands, they stand up. So what happens then, we pretty much using gel for changing our hairstyle. Let's see what we can actually do with this approach. As chemists, as material scientists, we can do many things. We can either change the hair, we can use different products on our hair, we can make them hydrophobic, hydrophilic, put something on their tips, or we can change the styling gel. And there are muscles that we can develop that respond to pH, to temperature, to different chemicals, to light. And that would put the materials in motion. And the outcome of that is just, let's take a look how these high aspect ratio materials put in motion. They stand up, they lie down. But when we want to use them, so what can we do with these structures? This example tells you that we can create fabrics that are super hydrophobic when they're standing up, which means in the rain, nothing gets wet. But on a dry day, when you want moisture to actually to go through your the materials that you wear, it becomes switchably hydrophobic or hydrophilic. You can actually use them, and we do use them, as a way to change our windows, that depending on the temperature outside, it will either let the light through or stand up and change the amount of light coming through, or simply create materials that change color or pattern in response to the stimulus. Yet another example shown here, you can use it to hide messages. Because only when they respond and bend, certain messages become readable, and they're not such when they're standing up and away from each other to interact. Okay. 2 more slides, maybe. I already see the time flies. Chapter 4, I want to talk about tulips, but specifically this Queen of the Night tulip. These iridescent seeds of Margaritaria nobilis, this blue color is not a dye. It is a structural color, such the same way as Morpho butterfly has it. In other words, and we probably all know that opals don't have a dye in it at all. And the structure of opal, it's composed of glass. It's S-iotospheres that are, depending on their size and depending how they pack, they form an array, a photonic crystal, that the color will depend on this packing. So that is the case for the ones that I've shown here. And this is just to show you another butterfly, Sesotris butterfly. This green, beautiful, beautiful green patch is structural color. It's actually what is called inverse opal structure. So where there were spheres, now empty spaces, we can make them. We can make them and have this structural color also made out of glass in this case. But we remember that butterfly, if it's not water, then it can infiltrate the structure. It's not used by butterfly for interesting applications or interesting functions. But I want to show you another organism. I've done a lot of work on this one. This is a relative of a star fish, and this is what is called brittle star, that actually changes color during the day and during the night. It was supposed to be, at least in the literature when I started to work on it, said that it's a biomimicry approach. No, it's not mimicry, because then you would expect that this creature would be white during the day and black during the night, so that to avoid predators. So it's exactly opposite of biomimicry approach, and what appeared to be the case is that, in fact, it has porous network, that when pigment comes and covers the lenses during the day and withdrawn back during the night. It's active optical system where liquid changes optical properties. In fact, what is it doing? Just really is sunglasses that change to be sunglasses or not to, depending on availability of light. So this is interesting, but can we now combine the lessons from these two organisms and think about dynamic optical devices? What if we use different chemistries in our colloidal crystal and functionalize them with different molecules that be more or less hydrophobic or liquid phobic? And therefore, I can encrypt any messages I want. Here's one nice demonstration. I've done a lot of work with marine creatures. Where liquid comes in, the structure is filled, the color is gone. So you can have a whole range of materials. This is, by the way, money was coming from Air Force, and this is butterfly that we've done it to it, and it says U.S. Air Force, and then it goes back when the liquid is evaporated. So what you can do, you can have rapid identification of liquids. You can use it for medical sensing. In particular, we've shown how to apply it in diagnosis of neonatal jaundice. There are many other things that you can do, or you can just do it for fun. And in particular, we had design students using it and showing how that can be used in your bathroom. When you take a shower, a lot of nice patterns appear, but the moment it dries out, it all disappears, and it really nice tiles in your bathroom that respond to environment because they're now sprayed with water, and only the places where water cannot come in remain colored. Here's a Harvard sign with, and this is a wonderful project when students decided to have a chess play. That color depends on the size of your pores, but to know exactly which piece this is, you have to put a specific liquid in it, and you need to know which one is that. So I will finish with showing last example, and this would be deep sea sponge that is called Venus's flower basket. Has nothing to do with flowers, but I really wanted to show this example as well. Its name is Venus's flower basket, but this is actually deep sea sponge that lives deep in the ocean, and it has in its structure one of the best optical fibers that we know, that are pretty much better than many of optical fibers that we make, but it's made of glass. It's one of the strongest glasses I know. In particular, I was even able to teach almost entire class in mechanical engineering based on the principles that this sponge is using in designing of its skeleton. And what is interesting, that in fact it is an illuminated glass house of the deep, because not only it resembles, and it's really strong material that resembles architectures that we use now. It's about a different length scale a little bit. But it also has a fiber optical network in it. And in fact, it is used by a pair of shrimp that lives inside the sponge, that takes advantage of the optical network that is connected to the ocean floor, taking light from the bioluminescent bacteria. In this way, it is a shining beacon deep in the sea, so food is attracted to it. And deep in the ocean, it creates really optimized materials for strength, fiber optics, and so on. So I will not talk about this. I just want to finish with talking about these principles. I want to say, let's all, especially in these difficult times, let's go and smell the flowers. I can, in addition to that, discuss with you which principles of smelling we need to take into account in our research if we design chemical sensors. But instead, I will summarize saying, let's do it together. We do need to bring together different sciences, different approaches to understand and principles which we can combine and create new materials. And let's not forget, we do not have to mimic one organism at a time. A lot of interesting strategies can come up by bringing together sometimes orthogonal strategies that are developed by different organisms, and in this way we can design really interesting materials. At the end, I want to thank my group. It's really eclectic group from every science possible in my students, and I want to finish with that. I hope that with our approaches we can keep it clean and not contaminated.
Well done. Thank you. Well, I know Dr. Aizenberg would enjoy answering questions. They should be short, and please say your name and where you're from. Thank you. There's one here in the back.
Rich Schiffer, Indiana University. That was a great talk. You gave wonderful examples of biology helping you design materials. How about materials helping you design biology?
I hope that can be done as well. Probably not in my lab, but we do use, in fact, I've done in my PhD, I've done a lot of work in using different biological molecules to change the properties of crystals and so on. So I think both are entitled to help each other, and I'm not really sure where there would be more help, coming from biology to help material science and engineering, or the other way around. I think both fields, or many other fields, can benefit from this two-way street.
Other questions?
Meadows, Philadelphia. I never knew so much about the structure and function of materials, but I'm wondering, these changes that come about that are functional and structural, they're controlled by genes, aren't they? They're genetic changes.
That's true.
Is anybody looking at the genes that make these changes possible?
Absolutely. In most cases, in most interesting, especially characteristic systems that are studied in biology, we are looking what is genetic makeup, which proteins, what is from the biology side that drives these materials, how they evolve and what is happening to them. What I was trying to say, that we can reduce the complexity to a manageable set of principles that are not exactly the same as biology. That's why I would rather call it bio-inspired than biomimetic, not mimicking nature by taking lessons and reformulating them to use different materials, and instead of genes, to have other ways of encoding information. And we have the ways to encode information. We also have the ability to use different materials. Nature is actually quite limited in the repertoire of materials it has. We can use steel for something that doesn't exist in nature, but it brings yet another level that we can use. But key feature is to extract manageable set of lessons that can be reformulated to the function or to the type of application that you have in mind.
All right, I think with that we'll go on, and let's thank Joanna once again for beautiful talk.